Okay, let's unpack this. We are diving deep into anabolism.
Right. The phenomenal energy-intensive process by which living cells, especially microbes, build order out of chaos
It's really how a single bacterium goes from just a few simple molecular pieces.
to a fully functioning, complex organism.
Yeah.
That creation of order is the critical principle here.
Hmm.
Anabolism is inherently a battle against entropy.
Which means it needs energy. Lots of it.
Immense energy. And we're not just talking about growing cells either. Even a non-growing cell spends serious energy just maintaining itself through what we call molecular turnover.
Constantly breaking down and rebuilding.
Exactly.
And what really brings this home, I think, is the antibiotic story mentioned in the sources. Penicillin, streptomycin.
Yeah, the discovery timeline is well-known Fleming, then Shane and Florey, Waxman coining the term antibiotic.
But the why is fascinating from a metabolic standpoint.
Totally. Why would a microbe spend all that material and, high-grade energy to synthesize such a complicated molecule, just to mess with a competitor?
It really shows the power of these anabolic pathways. They're master builders.
Incredible architects, really.
So our mission today is to give you the ultimate shortcut to understanding that mastery. We're going to look at the core principles, the building blocks,
Correct them from simple skeletons right up to complex things like membranes.
and even explore some really ingenious molecular delivery systems. Think of this as your distilled guide to how microbes construct life itself. So let's start with the basics. Cellular construction isn't random. There's a logic, a blueprint for efficiency. The sources lay out about 6 key principles.
Yeah, 6 sort of laws of cellular efficiency. The first one is fundamental. Large molecules are built from small monomers.
Like Legos, right?
Exactly like Legos. Macromolecules are built from simple structural units linked over and over. It saves a ton of genetic storage space.
And makes the whole process repeatable and, I guess, less prone to errors.
And less energy costly overall, yeah.
Yeah.
Very efficient.
And then jumping ahead slightly to maybe the 6th principle, these large molecules often know how to assemble themselves.
That's the beauty of self-assembly. The information for building larger structures, like ribosomes or even flagella, is inherent in the shape and chemistry of the parts.
They just snap together.
Pretty much, spontaneously. No extra enzyme needed just to stick the final pieces together.
Okay, back to resource management. You mentioned amphibolic pathways.
Right. Many enzymes do double duty. They work in catabolism, breaking things down, and anabolism, building things up. Saves resources.
But wait, that sounds tricky. If you share the tools, how do you control building versus demolition? If you stop one...
Don't you stop the other. That's the key question. And the cell's solution is elegant.
Okay.
While many steps are shared, the really crucial control points those irreversible reactions that act like valves controlling the flow. They use separate dedicated enzymes for catabolism versus anabolism.
Ah, so different enzymes for the key regulatory steps.
Precisely. This allows for independent regulation. And that's vital because end product regulation, where the final thing being built shuts down its own production line, is super important in anabolism.
Got it. Which brings us to energy. Anabolism needs energy input. It's endergonic. Principle 3.
Yep, biosynthesis has to be coupled to energy release, usually from ATP hydrolysis. That's the main currency.
But what about the electrons needed for building, reducing power?
Good point. That's another layer of separation. Catabolism, breaking things down, usually generates NADH, which is great for making ATP.
Right.
But biosynthesis, building things up, typically uses NADPH as the electron donor.
So different carriers for energy currency and building blocks, essentially.
Different electron carriers, NADH for ATP, NADPH for building. It's another control mechanism.
And that fits with principle 4, compartmentation, keeping things physically separate.
Exactly. Easy to see in eukaryotes with their organelles like mitochondria and chloroplasts.
But bacteria manage it too.
They do, yeah. Think of things like carboxysomes, little protein shells that concentrate the CO2-fixing enzymes away from the rest of the cytoplasm. It allows pathways to run side by side without interfering.
Okay, so the cell has these efficiency rules. Where do the building materials actually come from?
It all starts with just 12 core precursor metabolites. These are simple carbon skeletons, mostly intermediates from glycolysis and the TCA cycle.
Only 12? That's amazing.
It's a beautifully limited palette. From those 12, the cell makes everything else, all the amino acids, nucleotides, lipids, sugars.
If you're a heterotroph, like us, you get these precursors from food. Easy enough.
Relatively easy, yeah. But if you're an autotroph, a plant, or many microbes, you've got the massive challenge of making those organic skeletons from scratch. using inorganic CO2.
Carbon fixation. And that's energetically expensive.
Hugely expensive. You're taking a very stable oxidized molecule, CO2, and forcing electrons onto it to make reduced organic carbon. That takes serious energy input.
The main pathway for this, in aerobic autotrophs anyway, is the Calvin-Benson cycle.
Correct. The Calvin cycle. You can think of it in 3 phases or acts.
Okay, Act 4.
Carboxylation. The famous enzyme Rubisco grabs a CO2 molecule and attaches it to a 5-carbon starter molecule, RoBP.
Act 2.
Reduction. The resulting molecules get reduced using ATP and, crucially, that NADPH reducing power we mentioned. This makes the actual building block sugars.
And Act 3 is just getting back to the start.
Regeneration, yeah. Rearranging molecules to remake the RoBP starter, which also costs ATP.
So what's the bottom line? The cost.
Okay, here's where it gets really interesting, the metabolic price tag. To fix just 1 molecule of CO2,
Yeah.
the cycle burns through 3 ATP and 2 NAG-DPH.
Wow. Okay, so to make 1 glucose, which has 6 carbons.
You need to run the cycle 6 times, so that's 6 CO2, 18 ATP, and 12 NADPH.
18 ATP and 12 NADPH for 1 glucose.
It's an astronomical energy investment. Really highlights why autotrophs like photosynthetic bacteria or algae need such powerful energy harvesting systems just to break even metabolically.
Which probably explains why there are other ways to fix carbon, too.
Exactly. What's fascinating here is the diversity. There are at least 5 other known CO2 fixation pathways, like the reductive TCA cycle or the reductive acetyl-CoA pathway.
Different solutions for different microbes or environments.
Precisely. Suggests adaptation to specific niches, maybe where the Calvin cycle isn't ideal, perhaps because of oxygen sensitivity or different energy constraints. Life finds a way.
All right, moving from fixing carbon to actually building bigger things. Carbohydrates first. If a cell needs glucose, but doesn't have any,
It runs gluconeogenesis, basically making glucose from non-carbohydrate precursors like amino acids or lactate.
is it just glycolysis run backwards?
Not exactly. That's a common misconception. It shares, I think, 6 enzymes with glycolysis, the Embden-Meyerhof pathway.
Okay.
But 4 key steps, the irreversible ones in glycolysis, need completely different specialized enzymes in gluconeogenesis.
Ah, there's that independent regulation again. Separation equals control.
You got it. And when the cell needs to build more complex sugars, like polysaccharides or components of the cell wall, it uses special carrier molecules.
Like molecular delivery trucks.
That's a great analogy. Just like ATP carries energy in its phosphate bonds, these carriers ferry sugars around. The big one is uridine diphosphate, UDP.
UDP glucose, UDP galactose, those kinds of things.
Exactly. UDP carries the sugar unit to where it's needed.
Yeah.
And for making storage polymers like starch or glycogen, cells often use ADP glucose, so adenosine diphosphate, carrying the glucose.
These carriers must be crucial for building really complex structures, like the bacterial cell wall, peptidoglycan.
Absolutely essential. Peptidoglycan synthesis is, well, it's a masterpiece of spatial and chemical coordination. It happens across 3 distinct cellular locations.
3. Okay, where?
It starts inside, in the cytoplasm. That's where UDP is used to make the NMO and NAG sugar precursors, and the little peptide chain gets added to NM.
Step 1. Cytoplasm.
Then
Mirror?
the whole unit, the NAG-NAM-pentapeptide part, gets handed off to a different carrier. A huge 55-carbon long lipid embedded in the plasma membrane called bactoprenol.
Whoa, 55 carbons. That's massive.
It is. Think of bactoprenol as a specialized ferry boat anchored in the membrane. The NAG sugar gets added to the NAM-peptide on this ferry, forming what's called lipid 2.
So step 2, assembly on the bactoprenol ferry at the inner membrane. Where does it go next?
The ferry needs to flip. The whole lipid second unit has to be transported across the plasma membrane to the outside face. This flipping is done by an enzyme called the MERJ flipase.
Okay, flip to the outside into the periplasmic space. Step 3.
Now we're in the periplasm, the construction site. The NAM-NAG unit is added to the growing peptidoglycan chain.
But the wall needs cross-links for strength, right?
Exactly. That's the final critical step. Transpeptidation. Enzymes called transpeptidases, which are also known as penicillin-binding proteins or PBPs, stitch the peptide side chains together.
They weld the chains.
They do. They usually snip off the very last amino acid, a D-alanine, from one peptide chain, and use that energy to form a new peptide bond with a neighboring chain.
Yeah.
That creates the strong mesh-like structure.
And that's where penicillin comes in.
Precisely. Penicillin and related antibiotics directly bind to and inhibit those PBP enzymes. They stop the final welding step.
So the wall can't be properly cross-linked and it becomes weak.
Right. The cell tries to grow, pressure builds up, and without a strong wall, pop, it lyses.
Makes sense. And you mentioned another antibiotic, bacitracin, the stuff in Neosporin.
Yeah, bacitracin works differently, but it's also involved here. It interferes with the recycling of that bactoprenol ferry.
Ah, so it traps the fairy?
It prevents bactoprenol from getting ready to pick up a new NAM-NAG unit inside the cell. So it blocks the supply line of building blocks to the outside. Very clever.
Okay, besides carbon and sugars, cells need other elements integrated. Nitrogen and sulfur are key. How does nitrogen get in?
It usually starts with ammonia, NH3. If there's plenty of ammonia around, it's relatively straightforward. An enzyme called glutamate dehydrogenase can directly add it to a carbon skeleton, making the amino acid glutamate. That's reductive amination.
Simple enough. But what if ammonia is scarce?
Then the cell uses a much more efficient two-enzyme system called the GS-GOGAT system. It has a higher affinity for ammonia, so it can scavenge it even at low concentrations. It costs a bit more energy, though.
And once that first nitrogen is incorporated into glutamate or glutamine?
Then enzymes called transaminases take over. They basically shuffle that amino group, NH2, from glutamate onto various other carbon skeletons, those precursor metabolites we talked about, to make all the different amino acids the cell needs.
Now, you mentioned needing to be careful with terminology around reduction for these elements.
Yes, this is really important. For both nitrogen and sulfur, we have to distinguish assimilatory reduction from dissimilatory reduction.
Assimilatory means?
The element, like nitrate, NO3, or sulfate, SO4, is reduced specifically so it can be assimilated or incorporated into the cell's own organic molecules, amino acids, nucleotides, etc., building biomass.
Okay, building itself. And dissimilatory?
That's completely different. In dissimilatory reduction, the microbe uses nitrate or sulfate, not as building material, but as an electron acceptor for respiration, usually when oxygen isn't available.
Like anaerobic respiration.
Exactly. The end products aren't incorporated. Their waste products are released into the environment, like nitrogen gas, N2, nitrous oxide, N2O, or hydrogen sulfide, H2S. Functionally opposite processes, even though they both involve reduction.
Got it. Assimilatory builds the cell. Dissimilatory is for breathing without oxygen. What about fixing nitrogen gas, N2?
Nitrogen fixation. That's the ultimate assimilation challenge. Taking inert N2 gas from the atmosphere and converting it into ammonia, NH3. Only certain bacteria and archaea can do this.
Using the nitrogenase enzyme.
The incredible nitrogenase enzyme complex, yes. It's a biochemical marvel. But it's incredibly energy expensive.
How expensive?
Get this. It takes at least 8 high-energy electrons and a minimum of 16 ATP molecules just to convert one molecule N2 into 2 molecules of ammonia.
16 ATP per N2.
Wow, it's staggering. And on top of that, the nitrogenase enzyme is extremely sensitive to oxygen. It gets destroyed by it.
So nitrogen-fixing microbes need special strategies to protect it.
They do. Thick slime layers, living in anaerobic nodules on plants, super fast respiration to consume oxygen, all sorts of tricks.
Okay, and sulfur assimilation.
Yeah.
Similar idea.
Similar pattern. Most microbes take up sulfate, SO4. For assimilatory sulfate reduction, the sulfate has to be activated first.
Activated how?
By attaching it to ATP and then to another part of the ATP molecule, forming a special carrier called PPS phosphodenosine, 5-phosphosulfate.
PPS, another carrier molecule.
Yep. Once it's on PPS, then it can be reduced down to sulfide, S or H2S, which is the form incorporated into amino acids like cysteine and methionine. And again, this is distinct from dissimilatory sulfate reduction used in anaerobic respiration.
All these pathways feeding amino acid synthesis, it seems complex to manage.
It is, but cells are efficient. They use branched pathways extensively. One precursor metabolite, like oxaloacetate from the TCA cycle, can be the starting point for a whole family of related amino acids, lysine, threonine, methionine, isoleucine.
Save steps, I imagine.
Exactly. But it also means you're constantly pulling intermediates out of central metabolism, like the TCA cycle.
Right. If you're making lots of amino acids from oxaloacetate, you could run out of it for the TCA cycle itself.
Precisely. So the cell needs ways to replenish those intermediates. These are called anaplerotic reactions, literally filling up reactions.
Like what?
A common one is using pyruvate carboxylase to convert pyruvate directly back to oxaloacetate. Or, under certain conditions, microbes can use the glyoxylate cycle, which is like a modified TCA cycle that bypasses the CO2 releasing steps, specifically to produce more 4-carbon precursors for biosynthesis.
Okay, moving on to the last major building projects. Nucleotides and lipids. How does phosphorus get in? That seems simpler.
It generally is, yeah. Phosphorus is usually taken up as inorganic phosphate, pi. It gets directly incorporated into ATP through substrate-level phosphorylation, oxidative phosphorylation, or photophosphorylation.
And if the phosphorus source is organic.
Then the cell uses enzymes called phosphatases to chop off the phosphate group, releasing free pi that can then be used. Pretty straightforward.
And that phosphate is obviously crucial for nucleotides, the building blocks of DNA and RNA.
Absolutely. We have the purines adenine A and guanine G with their two-ring structure, and the pyrimidines cytosine C, thymine T in DNA, and uracil U in RNA with a single ring.
And how they're built is different.
They're fundamentally different, which is quite interesting. For purines, A and G, the double-ring skeleton is actually assembled piece by piece directly onto the ribose 5-phosphate sugar molecule. It starts complex and stays complex.
Okay, built on the sugar. What about pyrimidines?
They do it the other way around. The single-ring structure is synthesized first for simpler precursors like aspartate and carbonyl phosphate. Then once the basic ring is complete, the ribose sugar is attached.
Build the ring, then add the sugar. Different strategies.
Yeah, probably reflects different evolutionary paths or chemical constraints. And once you have these ribonucleotides with ribose sugar, you need deoxyribonucleotides for DNA.
How are those made? Just remove an oxygen?
Essentially, yes. The ribose sugar is reduced to deoxyribose. This reaction often involves a small, important protein called thioredoxin, which acts as the reducing agent, donating the electrons.
Okay, finally, lipids, fats, and membranes.
Right. Fatty acid synthesis is a major process. It's carried out by a large enzyme complex, the fatty acid synthase.
And the growing fatty acid chain isn't just floating around.
No, it's attached to another carrier protein. This one's called the acyl carrier protein, or ACP. The intermediates are shuttled around on ACP as the chain gets built up, usually 2 carbons at a time from acetyl-CoA.
ACP for fatty acids. Got it. And those fatty acids then get attached
to a glycerol 3-phosphate backbone. Adding 2 fatty acids to glycerol 3-phosphate gives you phosphatidic acid. This is a key branch point.
Branch point for what?
From phosphatidic acid, the cell can make either triacylglycerol storage fats or phospholipids, the main components of membranes.
And making phospholipids involves another carrier.
It often does, yes. Similar to UDP-carrying sugars, cytidine diphosphate, CDP, is often used to activate the head group, or the diacylglycerol part, during phospholipid synthesis. Another example of these nucleotide carriers playing a key role.
Okay, this brings us to maybe the most complex structure of all, especially in gram-negative bacteria, lipopolysaccharide, LPS.
Ah, yes, LPS. It's an incredibly complex molecule. You've got the lipid A part, which anchors it in the outer membrane and is the toxic component.
The endotoxin.
Right, then the core polysaccharide, and then the long variable O antigen sticking out. It's huge, branched, and has both fatty acid, hydrophobic, and sugar hydrophilic components.
And it has to get from where it's made inside all the way to the outer surface of the outer membrane. That seems like a huge transport problem.
It is a massive transport problem. The final assembly happens on the inner leaflet of the inner membrane, and it needs to cross the periplasm and be inserted into the outer leaflet of the outer membrane.
How on earth does it do that, especially the hydrophobic lipid A part crossing the watery periplasm?
Here's where it gets really interesting. The cell employs a dedicated multi-protein transport system called the LPT pathway, LPT for lipopolysaccharide transport.
A whole pathway just for LPS.
Entire pathway. It's basically a protein bridge spanning the entire cell envelope from the inner membrane to the outer membrane, made up of 7 core LPT proteins, LPTA through G.
A protein bridge.
Wow. It's amazing molecular engineering. Think of it like a scaffold or maybe even a slide or a chute. Proteins like LPTA form a track across the periplasm. They have a specific structure, sometimes called a Bujelli roll, that creates a sort of hydrophobic groove.
To protect the lipid A?
Exactly. It chaperones the hydrophobic lipid A tail across the aqueous periplasm, passing it from one LPT protein to the next until LPTD and LPTE in the outer membrane insert it correctly into the outer leaflet. It's an incredibly sophisticated delivery system.
So we've covered a lot. From the basic principles of efficiency.
The 12 precursor metabolites, the high cost of fixing carbon in the Calvin cycle.
The intricate 3-location synthesis of peptidoglycan involving UDP and bactoprenol.
Assimilating nitrogen and sulfur, distinguishing assimilatory from dissimilatory processes, the huge cost of nitrogenase.
Building nucleotides differently for purines and pyrimidines, using ACP for fatty acids.
And capping it off with that amazing LPT pathway, the protein bridge for delivering LPS.
So what does this all mean when you pull back and look at the whole picture?
I think it shows incredible cellular logic focused on maximum efficiency and conservation. Life builds immense complexity, the entire cell, starting from just those 12 simple precursors.
And using a surprisingly small toolkit of key carrier molecules.
Exactly. ATP for energy currency, NADPH often for reducing power, UDP for sugars, CDP often for lipids, ACP for fatty acids, bactoprenol for peptidoglycan units, PAPS for sulfate. It's a system built on reusable modules and specific carriers.
Yeah.
Profound chemical engineering.
And maybe a final thought to leave our listeners with. If knowledge is most valuable when applied, think about that LPT pathway again.
That 7-protein bridge.
Yeah, an entire complex assembly dedicated solely to transporting one type of molecule, LPS, across one specific gap. What does the sheer amount of cellular resources dedicated to that one transport job tell you about how absolutely critical LPS and the integrity of that outer membrane barrier are for gram-negative bacterial survival?
It underscores its non-negotiable importance. A fascinating point.
Definitely something to think about. Thanks for helping us unpack all that today.
My pleasure. It's amazing stuff.